Hi there, my name is David Smith. I'd like to introduce you to my lab and talk briefly about some of the research we do in our lab and what we're passionate about. So, here we go. Have you ever looked under the hood of your car, and have you ever wanted to know how it works? This is how we think about human disease. And to look under the hood of disease, you have to be looking inside the cell and understand the molecular mechanisms that are involved in the cell that might be causing any disease process. So we're interested in two specific diseases, cancer and Alzheimer's disease. And it turns out that people that get cancer are interestingly predisposed to not get Alzheimer's disease, and vice versa. People who get Alzheimer's disease are unlikely to get cancer. So we're really interested, is there some common biological mechanism that might predispose a person to getting either or one of these diseases? And we were curious about understanding what molecular mechanism in the cell might be involved in that. So it turns out that protein degradation increases as cancers progress. So you need the ability to degrade more and more proteins in order for cancer to progress. But in Alzheimer's disease, we have a situation where protein degradation decreases as Alzheimer's progresses. And this is not only true of Alzheimer's, but it's also true of other neurodegenerative diseases as well. In the cancer case, if we inhibit protein degradation, we can actually treat those cancers, such as with Velcade. But in the case of Alzheimer's disease, if we or neurodegenerative diseases, if we inhibit protein degradation, we can actually cause these diseases to occur. In fact, in Alzheimer's disease, this is caused by cells that basically fill up with undegraded proteins that aren't degraded properly, and it causes those cells, those neurons, to die, which causes the disease. So what's the primary molecular mechanism involved in degrading proteins? And that's the proteasome. So the proteasome is this huge 2.4 megadalton molecular machine that degrades proteins through an ATP-dependent process whereby it unfolds them, injects them into its interior for degradation. What we're interested in is trying to understand all the different regulatory ways that there are to degrade proteins. So there are many regulatory caps that bind the proteasome, and we're interested in understanding how they function from a mechanistic perspective so that we can learn and design new drugs that we might be able to use to either inhibit the proteasome or activate the proteasome, depending on which disease we happen to be looking at. So what are some of the methods we use in our lab? We do a lot of enzymology, which of course requires a lot of protein purification to understand mechanism. We also do biophysics and spectroscopy to look at how enzymes are working. If you want to know how an enzyme is working, you also want to know what it looks like. So we do cryo-EM or cryo-electron microscopy to build atomic models of what proteins look like the proteasome. And for more biological applications, we do a lot of human cell culture, and we also use CRISPR to modify the genetics of C. elegans, which is a small nematode worm, for animal models of studying these diseases. So in brief, that's our lab. That's what we do. That's what we're passionate about. And any of us in the Smith lab, including myself, we would love to meet you and learn more about the things that you're interested in. We would be happy to tell you about the science that we do here. So please reach out to me, reach out to any of these people here that you see around in the hallways, and we look forward to meeting you in the coming weeks and months. Thank you. Bye-bye.